Hey guys, Mr. Hyatt here. This is part 2 of the AP Biology Chapter 9 lecture. We left off, we had just talked about electron transport, and we're ready to kind of dive in and get lost in the weeds a little bit. So we're going to use sets of cytochromes, which are iron-containing proteins, to pass electrons back and forth across the membrane. Sometimes we're going to pass them along the same side of the membrane. These cytochromes alternate between reduced and oxidized forms and pass the electrons down to the terminal acceptor that's O2 or oxygen. So don't get lost in the fact that these are cytochromes. It is important that they're iron containing, they're metallic. They have some metallic structure anyway, so they're good at transferring electrons. But the idea is we're going to pass these along. When I say these, pass electrons along until oxygen catches it and we turn it into water. So the function of electron transport is to convert NADH and FADH2, which came out of the last step, and convert that into ATP, and it happens in the inner folds of the mitochondria. So what you should notice is basically what our mitochondria is attempting to do is take the scraps of an earlier process and make something out of it. So we're going to take the leftovers from glycolysis, we're going to pull them into the mitochondria, and we're going to make some more ATP out of it. Then we're going to pull the leftovers from the Krebs cycle over to the membrane of the mitochondrion, and then we're going to use the electron transport chain to pull a little bit more energy out of it. So basically, our bodies, through the mitochondrion, have become really good at making energy from the trash of our energy processes. So each ATP is going to, good grief, our ATP yields are going to be 3 per NADH and 2 per FADH2. So we're going to get several ATP per round. The chemiosmotic hypothesis you've seen before, but we definitely didn't have the tools to understand it until now. Essentially, the electron transport chain, in the electron transport chain, energy is moved to pump hydrogens across the Christie membrane. So that's that internal membrane. Let me skip to this picture. Remember, this is what we're talking about when we say Christie. They're the folds inside the mitochondrion. ATP is generated as that. So we spend energy to pump the hydrogens out, and then they're going to diffuse across the membrane passively, and we're going to create ATP that way. So the energy that establishes the gradient is most of that comes from exergonic reactions, and it creates a ton of ATP. Okay, so here's how it works. There are some animations online. I think I said something on a later slide. You probably want to watch and see how this works. But the idea is we're going to pump electrons into the inner membrane space. Then they're going to diffuse across, I think I might have said electrons, I meant protons. We're going to ship these H pluses out into the intermembrane space, and then it's going to passively move across and turn the wheel. The wheel's going to create ATP just like a water wheel. Getting back to this picture, we see several of these proteins together in our complex. So you see we're pumping out, diffuses back across. When it diffuses across, it's going to use ATP synthase. It's going to turn that water wheel, and that's going to create ATP. Remember when we talked in Chapter 8, ACE means it's an enzyme, and synth is what it does, so it's going to be something that synthesizes ATP. Excuse me. Kind of already talked about that. Works like an ion pump in reverse or like a water wheel. I've said that a couple times. Here's a link. It's not going to work in the video, but you guys have access to this presentation on Canvas. There are some good animations on ATP synthase and electron transport. I would watch those if I were you. I posted Mr. Anderson's talk on this chapter, and I think I posted the Konica. I posted several lecture type of things over this chapter, but I didn't do a good job of posting a lot of these animations. So check them out. They're all over YouTube. Just Google ATP synthase, or maybe search that in the address bar in YouTube or search bar or whatever you want to call it. But there's lots of good stuff out there. Alcoholic fermentation is done by yeast, which is a kind of fungus. So this is sort of kind of how, if you grow up to make beer like Mr. Hyatt does, this is the science that you're taking advantage of. In this picture, we see fermentation vats where this process of fermentation, cellular respiration, is happening like crazy, and we're taking advantage of it as humans. With respect to alcoholic fermentation, it's only going to use glycolysis. What you'll see is it's a lot like lactic acid buildup in our bodies. So essentially it's going to use only glycolysis, and it's going to cause an incomplete oxidation. So there's still energy available in the products. That's what we call alcohol. Notice this does not require oxygen, which makes sense. It's anaerobic respiration. And it produces ATP when oxygen is not available. So here's a diagram that shows alcohol fermentation over here. The ethanol is the alcohol that is produced. We've got glycolysis that creates pyruvate. Pyruvate is going to be converted into acetaldehyde. And then here we've got the fermentation piece of it where it's going to create the alcohol. Lactic acid fermentation, same thing. The energy still left in the products is called lactic acid. That's what makes your legs burn when you're doing anaerobic or when you're doing exercise. So, excuse me, when you run out of oxygen, your lactic acid starts to build up in your cells, causes soreness and stiffness, causes your muscles to give out. We have to take lactate to the liver and convert it back into pyruvate. So that's why it takes a while for that feeling to go away. Same kind of process as what we saw in alcoholic fermentation, but this time we've got a different product. We've got lactate being made as opposed to ethanol. Slightly different process. Same kind of idea though. Fermentation is a way of using up NADH so that glycolysis can still run. It's kind of like an emergency system. There's no oxygen, so are we dead, or do we have an emergency backup system? Here's kind of a summary. In terms of glycolysis, we're going to create pyruvate. If we have oxygen, then that pyruvate is going to go into the mitochondria. And if we don't have oxygen, then fermentation is going to happen in the mitochondria. And that's true whether we're talking about yeast or whether we're talking about humans. With respect to aerobic versus anaerobic, of course aerobic means with air, anaerobic means without air. And when we have aerobic respiration, I keep saying air, I mean oxygen. When we have oxygen, all three steps of respiration can happen. When we don't have oxygen, only glycolysis can happen. Now, of course, just like we have generalists and we have specialists, some organisms can only do respiration one way. Some can switch back and forth depending on whether oxygen is there or not. Those are called facultative anaerobes. Yeast is an example. So since yeast can do both, aerobic and anaerobic, they're facultative, which is the better process? If they can do both, which one are they going to do when they can choose? If we look at anaerobic respiration, glycolysis is only going to produce 2 ATPs per glucose, where aerobic we're going to produce essentially 36 molecules of ATP. And here's the math behind that. I've got another slide that will really wrap it all together. So, excuse me. Glycolysis is going to produce 2 ATPs and 2 NADHs. Then the Krebs cycle is going to produce 2 ATPs, 8 NADHs, and 2 FADH2s. Each of these NADHs is going to make 3 ATPs, so that's 30. And then each of these FADH2 is going to create 2, so that's 4 more. So if we do the math, add all that up, we get 38 ATPs per molecule of glucose. That's kind of like the perfect scenario, perfect efficiency. But we're going to use a little bit of energy to move, especially to move from glycolysis into the mitochondria. So our actual yield is about 36 ATP per molecule of glucose. So yeast would rather do aerobic respiration because it's going to produce a lot more bang for their buck. But if there's no oxygen alive, at least anaerobic respiration will keep them alive. And that tells us that if we want to produce alcohol, we have to keep the liquid as it ferments in an oxygen-free or an oxygen-limited environment, because we want that fermentation to happen. We want the alcohol to be produced. We don't want it to respire aerobically. So why is respiration important? Well, it's going to convert food into ATP. We need that ATP. And then, as you saw, we use materials in other cellular pathways, not just respiration. Some of these things are going to be used all over the place. Skip this slide for now. What that's trying to show is a lot of things go in and a lot of things come out, and we push all this food that we eat through our system and we create energy with it. Other importance, of course, alcohol industry, baking industry. Most breads use yeast to raise dough and things along those lines, so that's an application. Yeah, so there's a reason that wine and things like that don't get higher than 12 to 14% alcohol, because the alcohol is going to be toxic and kill the yeast. So it's definitely a balancing act. Swiss cheese are another example. The holes are bubbles of carbon dioxide that form from fermentation. So in summary, know the 3 main reactions, know the 4 required items for each, appreciate the importances. That's a pretty touchy-feely kind of a thing. Ignore this slide. So we will leave off with the summary. We'll worry about regulation at another time. So that's chapter 9.